So now coming back to pathway enrichment analysis and its requirements, we talked about gene lists. Now we're going to talk about pathways. So pathways and other gene function attributes are used in pathway enrichment analysis. There are lots of information available in databases. A good source of pathway information is gene ontology, biological processes, terms, also pathway databases like Reactome, which we'll learn about in the workshop. But there are many other types of annotations associated with genes. For instance, molecular function and cellular location, chromosome position, disease association, transcription factors that bind a set of genes, and many others. So these all represent ways of annotating genes, and you can represent many of these as sets of genes, like a set of genes with a given molecular function, like a protein kinase, and the so this information, so in this workshop, we're going to be mostly focused on pathways, but there is a lot of additional information about genes that we can use similarly to pathways in pathway enrichment analysis methods. Okay, so talking about pathways first, I'm going to talk about the gene ontology. The gene ontology, or GO, is a set of biological phrases or terms which are applied to genes, like protein kinase, apoptosis, or membrane. This gene ontology is also a dictionary, so each term has a definition associated with it, and it's actually quite useful for teaching because tens of thousands of biological terms have nice definitions in this ontology. I've mentioned the word ontology, gene ontology, and ontology is a formal system for describing knowledge. So usually ontologies have a couple of different features. So one feature is that terms are frequently related to each other, which I'll explain in a bit. You also have a standard way of naming terms. And more information about gene ontology is available at the project homepage. So gene ontology is structured in a hierarchy. The terms are these boxes here, and the relationships between terms are varied, but two examples are is-a and part-of relationships. So we might see, for instance, B-cell apoptosis is a type of apoptosis, which is a type of programmed cell death, which is a type of cell death, this is a type of physiological process, etc., all the way up to a very general part of the hierarchy. B-cell apoptosis is also part of B-cell homeostasis, which is a type of immune cell homeostasis. So you get the idea of how these different relationships here have different meanings, and the advantage of organizing all of these terms in this hierarchy is that it describes multiple levels of detail about gene function. Terms can have more than one parent or child, and that sometimes complicates gene ontology analysis, but it's just important to understand that. So gene ontology covers three different aspects of gene function. Cellular component branch or aspect of the ontology covers where genes are located in the cell. The molecular function branch or aspect covers molecular functions like enzyme functions, and the biological process aspect covers pathways like cell division or more general processes. So gene ontology is actually composed of two parts. The first part are terms, which I talked about just now. Go terms are added manually by editors at various different annotation groups worldwide. They're added by request, and experts help with major redevelopment. The gene ontology terms, there are many of them. There are over 44,000 terms currently in the ontology. This grows over time. So it is evolving, although over the past few years it's remained relatively stable. They've covered a lot of the known concepts in biology. The second part of gene ontology are annotations. So this is a particularly interesting part of gene ontology that we use for pathway enrichment analysis. This links terms to genes. So annotations take a term and they connect it to a gene using an association. Usually trained curators of genome databases do this. They're known as, these links between genes and terms in the ontology are known as gene associations or Go annotations. Multiple annotations are possible per gene, and frequently genes have many different terms associated with them. Some gene ontology annotation is created automatically without human review, and this tends to be lesser quality, and so it's important to understand that there's two types of annotation, which I'll explain more in a bit. So because of this hierarchy, and because multiple terms are associated, can be associated to a gene, a given gene, once it's associated to a term, automatically associates that gene to all of the other terms that are the parent of that term that we associate to. And so that's one way of generating a lot of additional terms that are associated with a gene. As I mentioned, there are two major sources of annotation: manual, which are curated by scientists, and these are very high quality, although they're time-consuming to create, so there are fewer of them relatively. Another type of manual annotation are computational analyses methods, which generate a lot of annotation automatically, but then the results are reviewed by people and filtered to just keep the ones that make sense. So electronic annotation is generally annotation derived without human validation. Frequently computational prediction, the accuracy here varies. Some computational methods are extremely accurate, like computational methods that predict transmembrane regions in protein sequences are more than 95% accurate, reaching 99% frequently. But others are lower quality. So in general, this is frequently treated as lower quality than the manual evidence sources. So a key point is to be aware of the annotation origin. We can understand the annotation origin because each annotation, when a term is linked to a gene, is associated with an evidence code. There are lots of different evidence codes, like the ones listed here. All of the ones in red are manual or human curated or reviewed, and the one that's not is called IEA, or inferred from electronic annotation. All major eukaryotic model organism species and human are covered by gene ontology annotations. Many bacterial and parasite species are covered, and new species annotations are always in development. There's a current list of official Go annotations that can be downloaded from the gene ontology website, and you can always map gene ontology terms by orthology from a close species to a new species that's not annotated. If you were working with a genome that's not available, does not have standard gene ontology annotations available. So just to spend more time on understanding the relationship of gene ontology to these different species. So one point to be aware of is that while gene ontology annotations are available for many species, not all species are covered at the same level. So some species have many more annotations than others. So, for instance, human has the most annotations. Orange here is non-experimental, and blue is experimental. So there's over 200,000 terms that are associated with genes that come from primary experimental sources. And if we look at mouse, there's quite a lot. Rat, there's quite a lot. But chicken doesn't have that many. It's mostly non-experimental, which might be computational sources. And you can see how some species have far fewer annotations. They also might have smaller genomes, but you can see the variability in the difference between experimental and non-experimental annotations, as well as the number of annotations. There are many contributing databases, just to list a few. Most model organism databases and major genome databases and protein databases like UniProt contribute a lot to the gene ontology project. Gene ontology has too many terms for some uses, so people don't do this very much anymore, but imagine you wanted to create a pie chart of gene functions to summarize at a very broad level your genes. You might only want to show a few high-level gene ontology terms, and so GoSlim is developed as an official reduced set of Go terms that are available in generic form or for plants or yeast. And you can map your genes to genes, you can map your Go terms to slim terms if you want a very compressed small set of generic terms. There are many software tools available that allow you to browse the gene ontology, browse gene associations, and use gene ontology in different types of analyses. The website that I recommend if you're interested in browsing the gene ontology and its annotations is called QuickGo from the European Bioinformatics Institute. It's a very nice website, and it gives you lots of information about gene ontology. Gene ontology, so you're aware, is one of many types of ontologies. Anyone can create an ontology themselves by standardizing terms and their relationships, and I just want to give you a sense that there's more than one type of ontology. Usually in pathway network analysis, we only come across the gene ontology, but occasionally we come across other ontologies like a tissue type ontology or cell type ontology. Okay, so that covers gene ontology. I next want to briefly cover pathway databases. So pathway databases store information about biological pathways in more detail than gene ontology. There are hundreds of pathway databases that exist. There's also databases of gene sets that are derived from pathways and large meta-databases like pathwaycommons.org, which collects major databases and unifies them into one single portal. We'll go over pathway databases in more detail and cover databases like Reactome in the workshop. Pathways, and you can derive pathway information from gene ontology biological processes, as well as pathway databases like Reactome that I mentioned. And I mentioned PathGuide as a way for you to go and browse pathway databases that exist. There's many other types of annotations that I mentioned. And you can get these usually from genome browsers. So one that I like is Ensemble. Ensemble is a genome browser that pulls information from many different sources. Another one that's great is EntreGene from the US NIH National Center for Biotechnology Information. If you're working with a model organism, those databases usually are the best sources of gene attributes for those genomes, and there are many others that we could discuss during lab time in the workshop. Just as an example of how to use Ensemble Biomart. So Ensemble is a genome database that stores lots of information about genes. Biomart is a tool, a query tool that Ensemble makes available to allow you convenient access to gene list annotation. First you select your genome, then you select some filters and select attributes to download, and then you can download that information. I'm going to give you a demo of Ensemble Biomart because I find that it's not very intuitive the first time you visit it, but just a few pointers can get you on the right track. So I can access Ensemble Biomart from the Ensemble homepage at the top here. I'm going to click on Biomart. Sometimes Biomart takes a while. You have to wait some number of half a minute sometimes for it to do its job. So the first thing we do is choose a database. Because I'm interested in genes, I'm going to choose Ensemble Genes database, version 100. Then I'm going to choose a data set. I'm interested in human, so I'm going to choose human genes. Once I choose that, a set of filters loads here, and I can click on filters, and I have to wait for it to finish loading. I can click on filters, and I can ask for all the genes that are in a given region, like all the genes on chromosome 1. If I want to click that, if I want to know that my query is recognized and properly created, I can click the count button here, and it will count up the genes that match my query. So in this case, there are 5,475 genes on chromosome 1. I'm going to uncheck that, and I'm going to ask for a specific set of genes. So I'm going to limit my genes to ones with gene symbols that are some ones that a couple that I'm going to add here. Again, just to check that all of my genes are recognized, I've entered 2 genes. I'm going to click count, and the count should be 2 out of all the genes that are in the database, and that is correct. So now that I've verified that Biomart recognizes the input that I've added in the filter section, I can then click on the attributes section. And there's lots of different attributes I can download. So I can download features of genes, like different types of identifiers. I can download information about gene structures, like the exon start and end positions. I can download sequence information, like the peptide region or the 5' UTR region. And I'm going to go back to features here and download, select to download the gene stable identifier, but I'm not going to download these other things. And I'm going to scroll down to external identifiers, and I'm going to ask for the Go term name and the evidence code, and also the gene symbol, HGNC symbol. When I'm finished selecting features, I can click results, and the results will show me an example result, and you can use this to make sure that the information that you've have requested is there in the right columns. So here I'm getting a lot of different gene ontology terms for TP53. And then I can download everything to a file in different formats. I can choose to only have unique results, because sometimes these results are duplicated. And when I'm finished, I press Go to download all the information, and then I can load it up in a spreadsheet and work with it. Okay, so let's get back to our presentation. That's Ensemble Biomart. So what have we learned? Pathways and other gene attributes in databases exist, and there are many different types. We can get pathway information from gene ontology and pathway databases. Gene ontology is a classification system and dictionary for biological concepts. There are many types of annotations contributed by different groups. We can allow more than one annotation term per gene. Some genomes are annotated better than others. Annotation comes from manual and electronic sources, and gene ontology can be simplified for certain uses using GoSlim. And many other gene attributes are available from genome databases like Ensemble. So coming back to the pathway analysis workflow, just to remind everyone that we've been, in this background lecture, we've been talking about the identifiers that we're using at these steps here, where we collect genomics data. The results will use some kind of gene identifier to label genes. We'll probably keep those during the normalization and scoring approach, and we'll probably keep those to create a gene list. But then we might clean our gene list to make sure that all the gene identifiers are correct in preparation for analyzing them with a more detailed workflow. So that's it for the lab, for this lecture. This lab is made available if you want to practice working with gene identifiers and attributes, learning about gProfiler and Biomart. You can use the integrated assignment gene list or your own gene list to play with the gConvert system at gProfiler, or use Ensemble Biomart. I highly recommend just familiarizing yourself with those tools because they're very useful. I use them regularly, a few times a week. Okay, so that is the end of our lecture. The main point, again, of this lecture was to bring everyone up to the same speed on some very basic concepts that that everyone will need for the main workshop. Thanks for your attention.